Hello, everyone. My name is Dürkop. I'm the CEO and co-founder of Novozyme, and today I'm going to talk about process modeling. So my talk will be entitled From Experiments, Data, Hybrid Models, and Digital Twins, Several Up and Downstream Success Stories, highlighting the benefits of using advanced process modeling. Before we start with this topic, I quickly want to introduce myself again, or the company I'm working for. So as said, we founded Novozyme. It's a spin-off from Boku University in Vienna. Boku University in Vienna has a department of biotechnology already since 30 years. And the last 10 years, we intensively focused on process modeling. And from this knowledge we gained in this field, basically we founded the company in 2019, mainly addressing early stage process development to speed up the development process by reducing the experimental effort. And during the talk, I will show you some highlights from our work. This is all published, so you will always find to the bottom right, there's a quote, which you can then go to the internet and basically search for this publication if you're more interested in more details, or you can also write me an email afterwards. Okay, so starting off, how can we basically, what do we need to have better bioprocesses? So first of all, we have to understand our bioprocesses better. So that means we have to describe them in a certain way, in a certain QBD approach, so that we basically better understand how input and output is connected. The second thing is basically that we need better process scaling. So you know this, basically when you do process upscale, you often have the issue that whatever you develop in the smaller scale, the ideal set points do not hold true in the larger scale. And the third thing, basically, what we want to do when we look into the future is we also want to have process control. So basically, we want to interact with our process. And in case that something goes wrong, we want to know which screws to turn in order to get the process in the right direction again. For example, to maintain a certain quality attributes or a certain titer or whatever, just to bring it back. And all of these three things, as you might notice, are modeling topics. So basically, if you use nice models, you can have a faster process understanding. You can use these models for process scale-up, and also you need models for process control. So that basically we at Novozyme, where we have the vision, is basically to create a hybrid modeling toolbox. And we'll go into detail afterwards what is hybrid modeling, but the idea is to have this hybrid modeling toolbox, and this basically is fed by experimental data. So you have to perform experiment in a certain way. You have to feed this information into the toolbox. The toolbox then learns from this information and basically gives the process engineer the understanding of how input and output is connected. That's the first thing. And as said afterwards, when we also want to use these models not only for understanding and for optimization, but also for control purposes, we also have to bring it back to the manufacturing environment where we use this type of models for process monitoring and control purposes. Today's talk, I will have 6 showcases for you, 4 upstream showcases and 2 downstream showcases, and they were basically all developed in this hybrid modeling toolbox. There will also be a link provided at the end where you can download this toolbox and can test it yourself on your own data to get an idea of how this works. Basically, it helps you to develop time-resolved bioprocess models. It gives you a certain process knowledge that helps you to understand how input and output is connected, and you can take these models for process optimization and, as said afterwards, for control purposes. And already quite some pharmaceutical companies use this tool to optimize their processes. Before I want to start with the modeling task, I want to highlight one really crucial thing, because we always encounter this, that a lot of people believe that the algorithms are the only important thing to drive valuable process model, but it's actually not. So the first thing before you start modeling, before you do anything, is questioning yourself, what do you want to do? So what is the whole modeling task for? Do you want to do process optimization? Do you want to have a model for process control? What is the purpose of all of this model? Based on that, you can then think about which experiments to perform. So what are the critical process parameters I'm looking at? You can do this with a risk analysis, for example, to investigate which of the factors are really important and which are less important. Then you think about which experiment to perform. Afterwards, you collect the data. And when I say collecting the data, we're collecting the offline and the online data, because there's value in both of them. And only after that step, we are then going in the direction of process modeling. So the third step then is process modeling. And it's not just about the algorithm, but it's really about the research question. What do you want to do? Then building the type of experiments, data, process modeling. And based on that, you can then use these these models in a digital twin environment where you can do in silico process simulation to say, okay, what would be my ideal process condition? And then you either go back to the lab and perform new experiments, or, oh, this is a little bit off, you take these models basically for scaling or for manufacturing, for visualization. You can take this type of models then for the error for the second for the green line here. When we talk about hybrid models, what does this mean? Basically, hybrid models bridge the gap between mechanistic understanding and data. So, as we said, we have to have a certain idea what we want to predict from our models. What is the output? In this case, we want to have critical quality attributes 1 and 2. This one should be predicted by the model, and we have some critical process parameters that fed into this model. And then we have a mixture of a black box, the machine learning part, artificial neural network, for example, and these artificial neural networks then predicts or estimates some rates that are then put into mechanistic understanding. So like mass balances, which then at the end predict the critical quality attributes. And by doing so, you have the advantage that you have a precise identification with a smaller amount of experiments. You can use this knowledge for process scale-up, and as said, you can use the same type of models for monitoring and control. And all of these showcases I will show you today in detail for up and downstream topics. When talking about hybrid model, what is the advantage of using hybrid model? So, for example, this is one of the first publications here I want to share with you today. The link is down there to the right. What you basically see here is we were interested into describing a design space, a three-dimensional design space in a 20-liter microbial fermentation, and we wanted to describe basically how the cells react to different inputs based on an ordinary DOE endpoint description, based on pure black box modeling, and based on hybrid modeling. And when we do so, and we compare it basically to predict the biomass and to predict the product at the end, you see here that the error for the response surface model with an ordinary OLS model is basically very accurate. It's in the range of the analytical error, and so is the hybrid model. But the hybrid model has the advantage that you model, in this case, the entire process from the beginning to the end. You also capture the process dynamics, and you incorporate knowledge. So this is a lot of advantages, and the error is in a very similar range. When you now look at the product, basically you see that the error coming from the OLS model is in the range of 20%, while the error from the hybrid model is in the range of 10%. The reason why is basically that with the hybrid model, we connect the product formation to the specific to the biomass. So the more we know how much biomass we have in our system, the better we can predict how much product is expected at the end point. Therefore, we get a better precision with this hybrid model over the other two. The one I did not mention so far is the pure black box model. So it basically also describes the whole process in a time-resolved manner, but the accuracy is not as high as from the hybrid model. And by doing so, you can do multi-dimensional optimization. So you basically get one model to predict or to describe as many output parameters you want to have. In this case here, we only have product and biomass, but you could also, for example, think about product quality. You could also model glucose propagation over time. All of these factors could be taken into account, and they are all in this single model that describes the whole process at once. Second, due to the fact that you have this time-resolved model, you can then think about advanced screening method like this intensified design of experiment approach. I will go into detail afterwards, but in a nutshell, basically it just helps you to significantly reduce your amount of experiments to screen a certain design space. And finally, the model structure, as I said, can directly be taken for monitoring and control, because you just have to put the input parameters in, and then you get a prediction for the output parameters you're basically interested in. So these are the 6 showcases I want to show you today. 4 upstream showcases and 2 downstream showcases. Starting directly with the upstream showcase, it's about model-based process optimization. So here the idea is to start with a certain amount of experiment, with a small amount of experiments, and then as quickly as possible screening the whole design space to find the optimum with as little experimental effort as possible. So as said, the idea is, okay, you have to think about what do I want to predict, what are the critical process parameters I want to vary. Then you start with an initial guess. You perform your experiment, you create your hybrid model, and then you go into this in silico simulator, which then basically simulates all of the possible combination in this design space. Based on that, the system tells you an optimum. So what is the best favorable condition you can have, and then you look basically if you already performed this experiment, or if it's a new optimum. When it's a new optimum, that means you have to perform this experiment because you don't know it from the data yet. And once you perform this experiment, you basically take it into account and put it again into the model structure. And then you're in this iterative loop, and you just iterate through the design space until the moment where you don't get a new optimum. So basically your tool says, okay, this is the condition which is favorable. And then if you look and you already performed this experiment, you know that you found the optimum. And by doing so, you can screen through the design space. And with this publication here, for example, we could reduce the experimental effort from 27 experiments to screen the whole design space down to 9 by using this model-based design of experimental approach. And you can see this here in detail, for example. So we started, as said, with 5 initial experiments, and then we just iterated through the design space along these orange dots, and then found the optimum. In this case, we modeled space-time yield, and you can see, for example, in the first iteration where we only have 5 experiments, you see that the model is already very accurately describing the biomass. You see this in green, but it's quite off for the product. So that means you take this experiment B into account into the next iteration, put it into the algorithm, and do the same thing again and again and again. And after iteration F, basically you finish this optimization and you found your optimum process condition. And by doing so, you can iterate through the design space. If you have the ability to perform campaigns, for example, that you can perform 4 experiments at a time because you have a task heap or you have an Ember system or something like that, then you can also think about what would be the 4 best conditions to learn as much as possible from the design space to iterate through your optimum to your optimum. So that is the first thing I wanted to share with you. Again, here there's the publication on there. Just follow the link and read it in detail if you're interested. So that's the first thing I wanted to show you here. When it's about process optimization, you don't have a lot of ideas about your process, and you just start with some initial experiments. The second showcase I want to share with you is about process characterization. So it's more about you have a defined design space you want to screen, and here you can use something like intensified design of experiments. How does this basically work? You have several approaches how you can screen a design space. The most easy, so let's say the obvious one is to just perform one of the experiments that are possible. If you have a very large design space or something above 4 factors, this might turn infeasible because you have to perform that many experiments that you just cannot, you will never finish it. The second one is the obvious one is then you reduce your experimental effort by just taking out experiments. You see this in this fractional approach, where you just reduce experimental effort by just taking points out. But it's also quite clear that this comes with the drawback that you're not able to describe the design space in the same way because you significantly reduce the amount of experiments. And the third one is the approach I want to suggest here is this intensified design of experiments approach. What you do is you start with a certain parameter combination. So let's say to the corner left, and then during your cultivation, you change your process parameter combination, let's say to the center point here. And after a certain time again, you change this parameter combination again. And by doing so, it's quite obvious that you can basically put in this case, put 3 experimental parameter combination to 1 experiment. So you see that you only perform 9 experiments overall, but you pack 27 parameter combinations into a single experiment, pack 3 parameter combinations into each experiment. That means you can screen 27 parameter combinations with only 9 experiments. And now when looking at the results, basically you see this here. So we created a hybrid model based on this 9 intensified design of experiment runs, which are these orange dots, and created the hybrid model. And then we took this model and applied it to the static design of experimental approach, which is basically these triangles, these green triangles. And you see this is a prediction of the model over the measured values. And you see when you create the hybrid model in this 9 iDOE runs, yeah, for sure it's quite precise because it's trained on this data. But then when you take basically the static DOE data, so the runs where you do not change any parameter combinations, the classical approach, let's say it this way, you see that the hybrid model is able to predict the outcome of the static runs quite accurately. So in a nutshell, you can think about, okay, either you perform this 27 experiments or 31 because we did some replicates, or you perform 9 experiments to screen the design space. And it's quite obvious that the amount of work you have to put into 9 experiments is significantly less compared to the 31 experiments in the static. And you see to the left is scatter plot, and in the center on the right there are 3 runs, for example, as an example for static experiments with different outcomes and different timings. And you can see how the hybrid model is able to predict this outcome quite accurately, with the advantage that you save almost 70% of experimental effort. And second, which is maybe not so obvious when you think about model predictive control approaches, where you want to change process parameter combination in order to recover your process, you have to know basically how your cells react to process changes. And somehow, by applying this already during the learning, your model somehow understands how quickly basically process parameter changes lead to a certain quality change in your responses. The third showcase I want to share with you is a process scale-up modeling here. In this case, we took a CHO culture, and the idea was to apply a model from shake flask experiments, also to train a model on shake flask experiments, and then to apply it on the 15-liter scale. So it's a factor of 50 scale-up, and we want to predict basically how good, or we want to apply the knowledge from the small scale on the larger scale to find the optimum process condition on the larger scale. And the only thing we had to do in order to be able to describe the full design space on the larger scale was to take a center point cultivation to recalibrate the model on the larger scale. And then it was able to predict the outcome for all of the different other parameter combinations. You can basically see this here. So you have again the scatter plot to the left side. Top left is basically the viable cell density, how accurate this is predicted for the shake flask experiment, and to the left bottom, basically it's the product. So you see hybrid model was trained on these runs quite accurately, and then it was predicting the outcome on the 15-liter scale. So it's a little bit less precise, but still it's, I would say, very good. And you see also that the model is now able to describe much higher product formations, for example, while to the left the maximum was 0.6 grams per liter only, to the right it was 1 gram per liter. And this was possible just because we did the recalibration of the center point and took this information into the hybrid model, and then it was able to describe this larger scale also quite accurately. The last upstream showcase here I want to show you is basically then when you have such a model, so we now created hybrid model, we now save the experimental effort on the larger scale. How can we now take these models for process monitoring and control purposes? We have a collaboration together with Siemens in Vienna, and what we basically did is we Siemens set up yeast cultivation in this bioreactor down there with a DOE setup, so to change pH and temperature. They also had some off-gas measurement. They performed this experiment, sent us the data. We created a hybrid model and sent them back basically the valuable information, so the packed hybrid model in a DLL file. So the DLL file then contains all of the knowledge of, okay, what is the outcome if you change one of these input factors, and then they put it into the sidepod environment, and we're able to basically monitor monitor the biomass concentration in real time during next cultivations. And this is basically depicted here. So you see, for example, how the biomass propagates over time in this yeast cultivation. There were also some offline measurements, but you see that the soft sensor was working quite accurately. And you can use such type of soft sensor for manufacturing purposes. For example, induce your cells always at a certain concentration to reliably always get a similar output at the end of your cultivation. And this is a typical application, is that for a soft sensor, and was implemented in the living lab in Siemens in Vienna. The next thing I want to share with you is a downstream showcase. So here is about ultrafiltration flux prediction. So what we want to do here is to predict basically the duration of an ultrafiltration process as accurately as possible. And the vision we had in mind basically was to say, okay, we want to have a process characterization of a certain membrane with a certain product. We want to know in the whole design space, by changing different parameter combinations, how quickly can we finish such a process and how does it propagate depending on the input parameter. So we had a set of product and membrane combination, and then we cleverly chose our design space when you remember what I told you in the beginning, when I said it's not only about the algorithm, it's also about the design space. This is really crucial. The experiments are as crucial as the modeling approach itself. So it's not just the algorithm that will solve the issue. It's really about thinking about which experiments you have to perform in order to get the maximum out. We often see from customers, also when we were at the university, I think a lot of you also have the same impression that it's rather starting soon with some experiments than thinking about what would be the ideal experiment. And the longer you think about how to perform the ideal experiments, the better your initial guess already will be. So I think this is a really underestimated parameter. So think about your experiments as much as possible before you start them, with respect to what you want to deliver at the end. Yeah. When the last thing we want to do here is basically to take these models. Then once we have the design space, we want to take these models for to predict the outcome of batch TFF, fed batch TFF, single pass temperature for filtration, and diafiltration, so that we have one model that is able to describe all of these 4 different unit operations or operational modes in a single model. When thinking about the fed batch model, basically what we did here is we changed the 3 parameter combinations we were interested in. It's the TMP, the cross flow, and the starting concentration of our target protein, and then we let an artificial neural network predict basically the flux at each parameter combination, and then we put this flux into a mass balance, and based on this mass balance, basically a new concentration was predicted after a certain time interval, and then we take this new concentration and basically put it back into the iterative loop. And by doing so, you can get a multi-step ahead prediction, and you can predict basically the duration of the process or the outcome of this process, depending on your input parameters. And this is a very simple but clever structure in order to model such a process. And when we now look at the results, we see here, for example, the duration of such a batch TFF or fed batch TFF. For you, maybe to recap, the difference between a batch and a fed batch TFF is a surge tank. So when you, for example, are in the manufacturing side and you have, let's say, a 5-liter tank or a 10-liter tank, but you have 20 liters to process, you need to put this remaining 10 liters somewhere, and this would be put into a surge tank. So that means it's something like a diafiltration, but you're diluting it with your original protein concentration. The consequence is that you see basically the shape of the profile is completely different. But due to this hybrid modeling approach, and due to the fact that we could take this knowledge and we know how this works and put it into the mechanistic equations, we are able to predict both different operational modes. And you see here, for example, 5 models for 5 different protein concentration. You see that the model and the predictions are quite accurate in predicting the duration of how long such a process would take as a function of the input parameters. And you see the same for the batch TFF, and you see the same for the fed batch TFF. Yeah. In the second step, or in the next step, basically, you can take the same type of model. So you can take the same training data to predict basically the duration or to predict the conversion factor of an SBTFF. So what is done here is basically that we take the same training data. We just have to change the mechanistic slightly in order to predict the conversion factor for different serial and parallel SBTFF systems. So it's the same training data, exactly the same training data, but what we learn here is basically you see the experimental data, so the test experimental data are these dots, and the model prediction is basically are these lines. So you see how the conversion rate, depending on 1, 2, 3 membranes, and the feed flow rate basically changes. And this is already done, as said, from the initial scouting from the data set initially. So you can really predict the conversion factor from a certain membrane combination based on very limited amount of training data. And this is the advantage, for example, if you have in the manufacturing side, these proteins are quite expensive. And for SBTFF development, you often have to consume a lot of these materials. So finding a way of how to predicting the duration easier or faster or earlier on is very helpful. And here it's based on a very limited amount of training data, which is then able to predict the conversion rate for all of these different membrane types. And then you can take this, for example, when you think about continuous bioproduction, you want to you have a continuous bioprocess and let's say something in the input changes, your model basically knows if you have a higher concentration, how do you have to adapt the feed flow, how do you have to adapt the pressure in order to get the same, to change the conversion rate in a favorable condition that you then get the same output at the end of your bioprocess. And this can easily be done with such a model. So this helps you to basically reduce development time. I said it saves drug substance because your training data is quite limited. You don't need that much data compared to an ordinary SPTFF development. You get much better process understanding because all of these curves are there, and then you see, okay, what is the change of how much do these curves change if I increase the starting concentration? How much does this change in order of the feed flow? So all of this is basically intrinsic in the model. And as said, on the long run, it helps you also to enable advanced process control, because when you know how these curves behave as a function of changes in the input, then you can revert it basically and take it for process control. The last thing here I want to show you is basically diafiltration. So all of the things shown before are already published. This is still under development, but you can see, for example, in diafiltration, one of the problems we often see is that the prediction of the pH, for example, or the prediction of certain materials inside the solution basically is not constant over time or over diafiltration volume. And if you now, for example, incorporate effects like the Donan effect into your mechanistic equations, then you are also able to describe this behavior. And then you already know how many diafiltration volume you would take in order to hit a certain pH or in order to hit a certain excipient concentration. And these models can also help you to achieve these goals. Yeah. Okay, so well, to summarize, basically everything I showed you so far is it's really important that you consider the whole workflow rather than just the modeling approach. It's really about thinking about what do you want to do with your models, what are the experiments I need to do, and then thinking about the modeling approach and not just putting the modeling approach in the first line. Nevertheless, hybrid models are a sound basis to create process model and digital twins, so to do in silico optimizations, what I showed you with this iDOE approach. And this then helps you significantly to save experimental effort, resources, manpower, etc., etc. You can take these hybrid models also for process scaling, and you can take these hybrid models then for process control. So while in the upstream, for example, you can use it for monitoring purposes quite easily. In the downstream, you could use, as I showed you with this filtration topic, you can take this type of models in order to understand or in order to change process parameters to get always the same concentration at the end of an SPTFF approach, for example. Finally, if you're interested into using this tool yourself, you can basically use your smartphone, just make a photo of this app. You can download this toolbox basically and start modeling for free with this tool in order to describe your process as much as possible or as best as possible and to save time. Finally, I'm at the end of my presentation. I want to thank you for your time. If you have any questions, you can always reach out to me on LinkedIn or just write me an email, and then we can have a discussion about if you have any questions, any modeling questions or any other questions. I'm happy to help you. Thank you very much for your time and have a great day. Bye-bye.